We start a very important topic, human cortex. Reason putting up this very simple topics is after passing out, we really go to the anatomy or read all these stuff, which are very nicely in university also. So I think it is.
Good morning, everyone. My topic for today's presentation is human cortex, and my moderator is Dr. Shobhit Garg. This is an overview of all the topics we will be going through today. First, let's talk about the neuron. The human brain contains approximately 10 raised to the power 11 nerve cells or neurons. There are 4 parts of neuron. First is the cell body or soma, which contains the nucleus and is considered the metabolic center of neuron. Second are the dendrites, which are processes that arise from the cell body, branch extensively, and serve as major recipient zones of input from other neurons. Then we have the axon. It's a single process that arises from specialized portion of cell body or axon hillock and conveys information to other neurons. Finally, we have the axon terminal, which are fine branches near the end of axon that form contacts or synapses, generally with dendrites or cell bodies of other neurons, release neurotransmitters, and provide a mechanism for interneuronal communication. There are 2 types of neurons. There are projection neurons and local circuit neurons or interneurons. Projection neurons have long axons and convey information from periphery to the brain, which are sensory neurons, from one brain region to another, or from brain to effector organs or motor neurons. Then we have the local circuit neurons or interneurons, which have short axons and process information within distinct regions of the brain. This is a diagram showing the major features of a typical neuron. Then we have the glial cells. In addition to neurons, brain contains several types of glial cells, which are at least 10 times more numerous than neurons. We have the oligodendrocyte, microglia, astrocyte, and Schwann cells. First, we have the fibrous astrocytes, which are located along the contact neuronal cell bodies, dendrites, and axons in gray matter, and form a complete lining around the external surfaces of CNS and around CNS blood vessels. Their function is maintenance of extracellular ionic environment, secretion of gliotransmitters and growth factors, and structural and metabolic support of neurons, along with regulation of blood flow. Then we have the protoplasmic astrocytes, which are organized along white matter tracts, end-feet contact blood vessels, ependymal axons, and nodes of Ranvier, and they have a function of support of axons in white matter. Then we have the oligodendrocytes, which are of 2 types. We have myelinating oligodendrocytes, which form myelin sheaths around CNS axons, and satellite oligodendrocytes, which surround CNS neuronal cell bodies. Myelinating oligodendrocytes have a function of myelination, while the function of satellite oligodendrocytes is unknown. Schwann cells form myelin sheaths around myelinated axons and ensheath unmyelinated axons. Their function is myelination, which is biochemical and structural support of myelinated and unmyelinated axons. Finally, we have the microglia, which are located along the gray and white matter of CNS and the function is scavenging and phagocytosis of debris after cell injury and death and secretion of cytokines. Then we come to the layers of human cortex. On the basis of light microscopy, cerebral cortex is divided into 6 layers. First, we have the plexiform layer or molecular layer, external granular layer, pyramidal cell layer, internal granular layer, ganglionic layer, and pleomorphic or multiform layer. The plexiform or molecular layer contains transverse fibers and some scattered neurons, while external granular contains mainly stellate neurons. Pyramidal layer contains pyramidal neurons and some stellate cells and basket cells. Internal granular layer contains stellate neurons and outer band of Bullard, while ganglionic contains giant pyramidal cells and inner band of Bullard. And finally, we have the multiform or polymorphic layer, which contains neurons of various sizes and shapes, and it merges with the white matter. Not all part of the cortex is 6-layered. The hippocampal cortex, which lies deep in the temporal lobe and has been implicated in the acquisition of declarative memory, has only 3 or 4 laminae. The hippocampal cortex is regarded as evolutionarily more primitive and hence is called archicortex to distinguish it from 6-layered neocortex. Then we have the paleocortex, which is presumably more primitive and generally has 3 layers and is found on the ventral surface of cerebral hemispheres. The functional significance of different laminae, neocortex, paleocortex, and archicortex is not known, although it seems likely the greater number of layers in neocortex reflects more complex information processing. This is a diagram which is showing the 6-layered neocortex in motor cortex and visual cortex. The 4-layered archicortex, which is present in hippocampus, and the 3-layered paleocortex or piriform cortex. In the agranular cortex, external and internal granular laminae are inconspicuous. This type of cortex is seen typically in motor areas, while in the granular cortex, the granular layers are highly developed, while pyramidal and ganglionic layers are poorly developed or absent. This type of cortex is seen most typically in sensory areas. Then we have 3 other intermediate types, which are frontal cortex, parietal cortex, and polar cortex. Then we talk about the neuroanatomy. In the early stages of human development, 3 primary vesicles can be identified in the neural tube: prosencephalon, mesencephalon, and rhombencephalon. Prosencephalon further divides to form telencephalon and diencephalon, while rhombencephalon divides into metencephalon and myelencephalon. The metencephalon forms the pons and cerebellum, while the melencephalon forms the medulla. The mesencephalon forms the midbrain. The diencephalon forms the hypothalamus, thalamus, and epithalamus, while the telencephalon forms the cerebral cortex, hippocampus, amygdala, and striatum. There are 4 parts of the cerebral cortex of each hemisphere, which is frontal, parietal, temporal, and occipital. This is a diagram showing the frontal lobe, sulci, and gyri. We have the superior frontal gyrus, middle frontal gyrus, and inferior frontal gyrus, along with superior frontal sulcus and inferior frontal sulcus. And we have the precentral gyrus. This is a diagram showing the sulci and gyri of parietal lobe. There is the postcentral gyrus, postcentral sulcus, superior parietal lobule, intraparietal sulcus, supramarginal gyrus, and angular gyrus. These are the sulci and gyri of occipital lobe. We have the intra-occipital sulcus, the inferior occipital sulcus, and the superior occipital gyrus, middle occipital gyrus, and inferior occipital gyrus. Finally, we have the sulci and gyri of temporal lobe. We have the superior temporal sulcus and the inferior temporal sulcus, and superior temporal gyrus, middle temporal gyrus, and inferior temporal gyrus. Talking about the prefrontal cortex, it can be divided into dorsolateral and ventrolateral regions. The dorsolateral prefrontal cortex is involved in manipulation of data during working memory, and the ventrolateral prefrontal cortex is involved in maintenance of information during working memory. primary motor area is the area 4 of Brodmann and is located mainly in the precentral gyrus on the supralateral surface of the hemisphere. When these areas are stimulated electrically, movements occur in various parts of the body. The body representation is upside down, that is inverted homunculus, and the area of cortex representing a part of the body is not proportional to the size of the part, but rather to the intricacy of movements in the region. Hence, relatively larger areas of cortex represent the hands and lips. This is a diagram showing the degree of representation of different muscles of body in the motor cortex. Then we have the premotor area, which is area 6 of Brodmann and is located just anterior to the motor area and is responsible for programming the intended movements and control of movements in progress. The frontal eye field is area 8 of Brodmann and is located in the middle frontal gyrus, and stimulation causes both eyes to move to the opposite side, that is conjugate movement. The motor speech area, area 44 and 45 of Brodmann, lies in the inferior frontal gyrus of dominant hemisphere, and injury to this area will cause motor aphasia. The primary somatosensory area, area 3, 1 and 2 of Brodmann, is located in the post-central gyrus, and like the motor area, the body representation is upside down, that is inverted homunculus, and the areas of cortex that receive sensation from a particular part of body is not proportional to the size of that part, but rather to the complexity of sensations received from it. Hence, digits, lips, and tongue have unusually large representation. Then we have the somatosensory association area, which are area 5 and 7 of Brodmann, which help in recognizing shape, size, and texture of objects. Then we have the sensory speech area, which are area 22, 39, and 40 of Brodmann, and they lie in the posterior part of superior and middle temporal gyri of language dominant hemisphere and are responsible for interpretation of speech. The visual areas are areas 17, 18, and 19 of Brodmann and are located in the occipital lobe. Area 18 and 19 are mainly responsible for interpretation of visual impulses. The auditory areas are areas 41, 42, and 22 of Brodmann. They are situated in the temporal lobe and receive the auditory radiation. This is a diagram showing the location of some of the areas of Brodmann on the supralateral aspect and on the medial aspect of cerebral hemisphere. The blood supply of cortex. The cortex is supplied by branches of anterior cerebral artery, middle cerebral artery, and posterior cerebral artery. This is a distribution of the anterior, posterior, middle cerebral artery on the supralateral surface of cerebral hemisphere. Around 2/3 of the supralateral surface is supplied by the middle cerebral artery. And this is a diagram showing the arteries which supply the middle part, and around 2/3 of it is supplied by the anterior cerebral artery. The association fibers in human cortex, the cortico-cortical connections comprise the majority of connections within the cerebral cortex and can be divided into intrinsic, which are connections within one cortical area, associational, connections between cortical areas of same human hemisphere, and callosal, which are connections between cortical areas of different hemispheres. The major connections on the neocortex. Input are received from other cortical regions, hippocampal information, amygdala, thalamus, and brainstem modulatory systems, while output is given to other cortical regions, hippocampal formation, amygdala, thalamus, caudate and putamen, brainstem, and spinal cord. This flowchart shows the connections of the association cortices. Inputs are received from corresponding cortical areas and non-corresponding cortical areas of the association cortex of the contralateral hemisphere. Inputs are also received from motor and premotor cortex and other primary and secondary sensory cortical regions. Then this information is processed and output is given to mediodorsal, lateral posterior, and pulvinar nucleus of thalamus, caudate and putamen, cerebellum, and other primary and secondary sensory cortical regions, and motor and premotor cortex. Then this information is this information is processed, and caudate putamen and cerebellum, they relay information to ventro-anterior and ventrolateral nucleus of thalamus, which further relays information to motor and premotor cortex, from where information is again received by the association cortices. And the brainstem modulatory inputs also relay information to other primary and secondary sensory cortical regions, which further relay information to the association cortices. Now, the distinctiveness of the human brain. Compared with brains of other primate species, the human brain is substantially greater in size, with certain areas expanded disproportionately. The prefrontal cortex has been estimated to occupy only 3.5% of the total cortical volume in cats and 11.5% in monkeys, but close to 30% of the much larger cortical volume of the human brain. Conversely, the relative representation of other regions is decreased in human brain. For example, the primary visual cortex accounts for only 1.5% of the total area of cerebral cortex in humans, but in monkeys, a much greater proportion, around 17% of cerebral cortex, is devoted to this region. In addition, the expansion and differentiation of human brain are associated with substantial differences in the organization of certain elements of neural circuitry. For example, compared with rodents, the dopaminergic innovation of human cerebral cortex is much more widespread and regionally specific. The primary motor cortex and certain posterior parietal regions receive a dense dopaminergic innovation in monkeys and humans, but these areas receive little dopamine input in rats. The human brain is also distinctive at the cellular levels, with greater relative percentages of certain neuronal and glial cells in cerebral cortex. For example, the relative proportion of GABAergic interneurons is approximately 15% in rodent cerebral cortex, but in humans, nearly 25% of all neurons in most cortical areas are GABAergic. Intralaminar and polarized astrocytes are found exclusively in humans, and human protoplasmic astrocytes are larger and have more processes than the rodent counterparts. Astrocyte to neuron ratio is 5-fold greater in humans compared to rodents, leading to greater astrocytic coverage of neurons and synapses, and thus possibly enhanced regulation of neurotransmission in human cerebral cortex. As we can see, the astrocyte to neuron ratio is 5 times more in human brain than in rodent brain. These are my references. Thank you.
Thank you, Dr. Rishi. So we have actually covered the various layers of the cortex, right from evolution into anatomy, each group functions, supply, and how the human brain is different from, and the connections of cortex with other brain regions, how it is different from other species. So I think we have two questions coming to that. So let's feel the support the how many of you posted?
Declarative memory, this is the type of memory which is stored in our brain and can be retrieved by an active process. That is, we have to think and to retrieve it.
Any other question? Arthur, any questions, comments? Usually. Asking whether opposite field contribution I whether it is a function and whether it is similar to the functions of frontal eye fields.
In the occipital lobe, it is
I think the occipital lobe is concerned with binocular vision, whereas binocular conjugate, which are defined by your frontal eye fields, and then there is an optic pathway is concerned with that. It's a different. I think it is only related to the reception part, occipital. Not sure whether you're saying that this is also involuntary. The occipital lobe has anything to do with the motor control. It's purely sensory integration of the fields. Common that you have to check.
Movement is more part of occipital. It can maybe it has some overriding function of teaching that ultimately eye movement is muscles, and the occipital lobe is has been more related with the sensory part. Sensory reflex, it can be there that when we talk about intermediate ocular PGR, one eye will move, one eye will not move. That is a deficit of a pathway which involves frontal eye field, and I think PPA and there are 2 or 3 more fibers involved. Occipital is purely giving a clear vision. On the backdrop of clear vision, your eyes won't move. That will be taken care of on that part. What is probe model? So what actually is the model? Explain this model just merely decision making. Parts sorting.
And this about the reaction time. Yeah, for example, if the word red is written, it is easy to say green rather than say red. The word. So, Wisconsin car sorting test
touch the gut and on the basis of that. So in this, like for example, in schizophrenia, difficulty in consideration, this concept cards out can also abstract and the attention concentration. There's a deficit.
Go back to first slide. First slide. So I wanted to doctor there. And then one of the sections in blood supply, you said it is middle, it's medial section. Any comments, Dr. Aston? Srinivasan?
I can understand the context and other areas, but better than other areas, I can understand.
The functions of parietal cannulus. Functions of parietal. I mentioned the functions of parietal. Parietal functions can be
So lesion of parietal lobe causes hemi-neglect syndrome.
which part of the parietal lobe? Right or left? Parietal lobe में dominant होता है. अच्छा मतलब left side के तो non-dominant parietal lobe. Right side. Non-dominant or dominant? Huh? Non-dominant. Either right side. Why not? And heavy neglect happens. But why not a dominant one? Left side parietal lobe lesion है उसमें heavy neglect क्यों नहीं होता? Explications. Why only selectively right side is involved? Because right side will control the attention, the visual spatial attention of both the fields, right and left. But left is only concerned with the right field. So what will happen? अगर left का lesion होगा, but because right is intact, it will not give you the deficit. Right will give you the deficit. Why it happens? They say it's some evolutionary kind of a thing, compensation by the brain. The left side को language dominance दिया है, तो right side को visual spatial dominance. All the functions which you do visual spatially are defined by a parietal lobe, predominantly right side. So if someone has right side parietal lobe lesion or tumor, I think then the person will not be able to execute its environment adequately. Any specific questions regarding the layers? Why we have discussed so much about the layers? 6 layers. And what is the importance of fourth layer? Talking a lot about fourth layer. It is generally it's concerned with only the relay of thalamus. We only go to the fourth layer. Fourth layer is purely concerned with the stellate cells layer. It's only concerned with the sensory reception in the human brain. And when you see a 5 and 6 layers, these have pyramidal cells, and they are concerned with the output. These are mainly motor outputs, the motor areas, and the putamen or striatum or basal ganglia. Interesting part of this cortex is, or this class is, that the human cortex here, it is only giving an output to your basal ganglia. It doesn't receive direct. Every connection, every brain area is directly connected except basal ganglia cerebellum, where when the reverse connections are via thalamus and other connections. This is a very interesting point. So possibly the extrapyramidal lesion when they happen, when they involve basal ganglia, because motor cortex will inhibit basal ganglia, keep it in check. So some lesion in motor cortex happens, the check goes off, and the person would initiate OC symptoms or extrapyramidal side effects or hits, and which are very difficult to correct subsequently. Shall we? क्यों किस तरह की और आप जब so aura is involved or occipital lobe. You see patient with complaint of seeing unformed visual stimulations, unformed images. But if it is a formed image, then association cortex are involved. And temporo-occipital junction is an association cortex. इसलिए temporal epilepsy में हमको चेहरे दिखेंगे, formed images. That is why that depends upon the functional lobe of individual areas. Temporal lobe is also a huge area. उसमें जो association cortex है, जहाँ जो involved होगा, वैसा aura function.
The symptoms of manifest on a nerve that are cortex. If the sensory one is there is epileptic activity. So the person could report sensory perception, so hallucinations, confusion state, manifestation, which area is getting stronger. Came very late. According to cycle, earlier on this cortex and the area, there was a lot of focus on that. And we studied also in detail, but now we know that the distribution is not as precise as there, because there is individual variation, even if some particular area is affected. So sometimes the contralateral area starts functioning, and sometimes the adjoining areas they take over. So it is difficult to say in an individual patient precisely what will be the outcome, what will be the deficit. Coarsely, we can definitely say, but precisely and certainly not in, say, like humor surgery, etc., where they would require this information, where like they don't want some motor deficit to happen. So this representation in the cortex is not precise. That is there. We still, because it's a jumble of neurons and in individual person, it is very difficult to precisely predict what will happen if some certain area is damaged. So that is there. But still the course, clinically we do find that symptoms and the region, the area matches. That is all.
I think what do you call this? Like frontal eye field, right side, frontal eye field, left side. Similar areas. Any technical term to see. Basic conjugate areas or basic homotypic regions. Homotypic region means similar regions in both the bodies. The whole point is, except these homotypic regions, as I said, cortex, there are no two regions in the brain which actually is. There is a lot of intra-individual differences also. No two individuals will also have similar exact copycat architecture in terms of number, character, So that is why intra-individual variations are also very, very high. And that is why human brain is very complex. And when we see this neural network, as I agree that the well-defined regions which was worked by Trotman, that has now it is coming to more standstill because of your connection fibers, callosal fibers, relation fibers, intracortical neurons, interneurons, and so that is why now they are now they have got DWI imaging, diffusion weighted imaging, diffusion tensor imaging, DTI imaging, where they will image the tracks. It's a specific kind of an imaging in MRI, and they will actually give you. This has been conducted by neurosurgeons, to see whether any fibers of tracks have been disrupted during their traumatic brain injury, and all of the people are working it in psychiatric disorders also. So things are moving ahead. I think we also need to catch up with the neurobiological enhancements which are happening now, and not basically shying away from reading all this. Merely knowing phenomenology won't do. I think we should be more versed about various very fast happening developments in neuroscience. So in context of psychiatry, it's very important. So I think this is the first step. Then we'll gradually design lectures, subsequently more about neurophysiology and neuroanatomy, other steps for circuits. But I think citing her first presentation, she came early, she informed me about the presentation, she showed her presentation very early, the changes which we suggested she inculcated them, and the presentation, the confidence, the slides. I think it was a good presentations. First presentation. We all learned with every presentation. She also getting. We have to work upon your question answers. Thank you. If you have questions. Only to know this is very perfect. Something you know. Yeah. उसका 50% डालिए निकल आपने
चिपका देना उसमें इंग्लिश में लगाया हुआ हिंदी का होना चाहिए
एक हिंदी का ये सिमिलर का सिंगल पेज का उसका लास्ट आपका बाइंड हो गया अभी बाइंड होने में गया है
भीड़ जाती है ना फिर सब लोग सोचते कि मेरा इफेक्ट करेगा